Greetings, my name is Sergio Dar, and I am presently a junior at Wayzata High School. My project title is CRISPR gene editing, removing antimicrobial resistance in bacteria. As advertised, my goal in this project was to try and find a method to mitigate the development of antibiotic resistance in specific bacteria. Specifically, I wanted to try and remove the mutations that are the root cause of antibiotic resistance in bacteria. Mutations can occur in any living organism containing DNA. We tend to either think of mutations as detrimental to our health or superpower-inducing, however, this is often not the case in reality. A relatively common example of mutations that are actually helpful can be seen in bacteria. Sometimes bacteria will develop mutations that create resistance to antibiotics. This greatly increases their survivability and therefore their ability to duplicate and grow. While this may be beneficial for the bacteria, in some cases it can be life-threatening for humans. If one such mutation occurs in a pathogenic strain of bacteria, it would become very difficult to relieve anyone that is hypothetically affected. These types of mutations typically occur when someone ingests antibiotics without a valid reason for doing so. This way, dormant bacteria will then gain exposure and develop immunity by mutating. While this is a valid point for prevention, getting rid of these superbacteria after they mutate is a much more difficult topic. With standard antibiotics out of the question, medical professionals are forced to rely on creating new cures and methods of treatment in response to the superbacteria. In order to remedy this issue, I considered the use of CRISPR, which is an acronym for Clustered Regularly Interspaced Palindromic Repeats. CRISPR is a gene editing tool that was discovered during 2012 in yogurt cultures, but it was further studied and introduced as a gene editing tool in 2014, which revolutionized modern gene therapy. The CRISPR process makes use of a programmable protein called Cas9. This protein has a rather simple yet important job. It locates a segment of DNA and cuts it out. What's so revolutionary about it is that it's possible to direct Cas9 to target a specific strand or gene using gRNA, which stands for guide ribonucleic acid. The Cas9 attaches to the location specified by the gRNA and cuts it out of the genome. You can then replace the DNA in that area by simply injecting in template strands of nucleotides. This essentially allows us to cut out the DNA at a specific location of your choosing in the genome and replace it, as long as you can create your own customized gRNA. By applying this to the superbacteria, we could possibly replace the gene causing the mutation entirely, ending the issue there and then. CRISPR is a very convenient tool as well, since Cas9 only targets the desired area. This one simple fact has completely revolutionized modern gene therapy, but for the purposes of this experiment, we can utilize it to only target the mutated area. This means that it would be completely harmless to the rest of the body and could be injected anywhere, since it would scan the entire genome for the mutation and the mutation only. However, it would be most efficient to target the location nearest the area of effect of the bacteria. My goal for this project was to recreate this in a lab-to-pilot experiment to determine if replacing mutations in bacteria is possible outside of theory. Prior to performing my experiment on this idea, I decided to utilize a non-pathogenic strain of Escherichia coli known as E. coli K12MG1655, which contains the beta-lactamase resistance gene known as AMP-C. This gene flushes out beta-lactam antibiotics outside of the bacteria cell like a pump before they're even allowed to act on the microbe. This drastically increases the survivability of the bacteria and makes it invulnerable to beta-lactam antibiotics, which can create huge problems in the medical field. Thus, my research question was, can CRISPR-Cas systems be utilized to remove antibiotic resistance genes in E. coli to combat beta-lactam antibiotic resistance? And my hypothesis was that using CRISPR-Cas systems to specifically target and disrupt AMP-C beta-lactamase genes in the specific strand of E. coli will restore beta-lactam antibiotic susceptibility and vulnerability. I used an online tool made by the National Institutes of Health to find the genetic coordinates of the AMP-C gene so that I could program the gRNA. After ordering gRNA specific to the targeted gene, I carried out my experiment. I began by pouring the bacteria into petri dishes containing agar, which is a nutritional environment for bacterial growth. I then added in a transformation mixture in order to create competent cells upon which I can easily inject CRISPR. After some period of incubation in a freezer, I took out half of the petri dishes and mixed them with the gRNA and the Cas9, leaving the other half as a control group. I then incubated all the samples again, and on the third day of the experiment, I took them out of the freezer and mixed them all with amoxicillin, which is a beta-lactam antibiotic. I then left them out at room temperature before returning to record results. After ordering gRNA specific to the targeted gene, I carried out my experiment. The first step is to prepare the plates. I started by microwaving the agar in a glass bottle for 10 minutes. Afterwards, it should cool for 20 to 30 minutes, and while the bottle is still warm, the agar must be poured onto the plates. Let the plates cool overnight in order to let the condensation evaporate, and the plates should be stored upside down inside a freezer, preferably at 4 degrees Celsius. The agar creates a nutritional environment for the bacteria to begin growing. The second step is to add the bacteria. In order to access the bacteria, I added 100 microliters of water to the bacteria tube and shook it for 1 minute to ensure the bacteria was fully dissolved. I then pipetted all 100 microliters of dissolved bacteria onto an agar plate. Using an inoculation loop, I then spread the bacteria onto the agar plate. After that, I made 3 tubes of bacterial transformation mix and bacterial solution for the CRISPR trials. I added in the bacterial transformation mix to each of the petri plates to ensure that I could create competent cells, upon which I can then easily inject the CRISPR. The third step is to begin the DNA transformation. I started by combining 10 microliters of the guide RNA with the Cas9 and the new template DNA that's going to be used to replace the previous DNA into this competent cell mixture. After that, the fourth step is to begin the incubation process. I started by incubating the tube at room temperature for 5 hours. This step allows the bacteria to recover and replicate the DNA, which is what happens when you actually perform the CRISPR engineering process. Lastly, incubate the plate at room temperature for about 1 to 2 days. The fifth and final step is to begin your experimental trials. I created 3 such plates of modified bacteria and grew 3 plates without the modification as a control variable. I poured the solution of all 6 plates containing the bacteria into a mixture of sterile water and 0.5 milliliters of amoxicillin, which is a beta-lactam antibiotic. I waited 24 hours before recording results and gathering data. I then concluded the experiment and reevaluated the process, as well as the implications of the results. The dishes with the unmodified bacteria had significantly greater numbers of CFUs, which stands for colony-forming units. This indicates that since the genetic modification was the only change between the strains that were tested, the CRISPR application was successful in completing the desired results. On a broader scale, this means that bacteria mutations can indeed be fought using the aid of CRISPR technology. Although more consideration is necessary if the mutation is potentially transferred to strains in nearby plasmids through horizontal transfer, our hypothesis, however, is still reinforced by our findings and data, and my conjecture is supported by the results of the trials at hand. Although the experiment was a success and proved our hypothesis to be correct with the data obtained, it is constrained by the materialistic limits on precision and accuracy. A simple but important way to improve this experiment would be to conduct more trials. Due to the heavy regulations surrounding some of the materials used in this experiment, this is not easily achievable for a student, but would be more possible for a researcher with the correct credentials. By conducting a greater number of trials, using different bacteria strains aside from E. coli and also different antibiotics, a much more thorough experiment could be conducted. On an even grander scale, you could change the gRNA itself and target an entirely different set of genomic coordinates altogether to test and experiment with what gene is the best to remove for the greatest amount of resulting effectiveness. Using a more powerful software instead of just human recognition to count the number of CFUs would be another great way of improving accuracy in this type of experiment. A secondary partner or helper to aid in ascertaining results and generally conducting the experiment would make the entire process much faster and less prone to errors, which would then result in a more accurate and precise data due to a reduction in human error. If ever conducting this on a similar experiment again, these steps should be incorporated into the procedures. Mutations could still spread into our modified bacteria through horizontal transfer if given the opportunity, but in this case, this is preventable with caution, and the temporary vulnerability is enough of a window to relieve a patient. CRISPR is a revolutionary gene editing tool with real potential for bringing about positive change for the environment and the medical field. It can be used to treat genetic diseases, create disease-resistant crops, and even fight bacterial mutations as supported by the evidence gathered in the experiment. Since it is still a relatively new technology, there is a lot of restrictions and regulations placed on it, but in the future, I can't even fathom to the extent which CRISPR may be able to bring about benefits for all of humanity. I believe that this experiment specifically has a positive real-life application with significant and positive benefits in future experimentation and exploration. Thank you for listening to my speech.